Hello, everybody. Welcome to the recorded version of the second lab in Biology 142. This lab will be mostly about the special sense of vision and a little bit about gustation and olfaction at the very end. To follow me along in this process, you need a lab report, and you will find this in a PDF format next to the video, the recording, and you will also need a set of PowerPoints that I will be showing. They will be attached in a PDF format right next to the lab report. So when we look at the special senses here in the lab, we have covered the special sense of hearing, and we have seen just how much effort the special sense of hearing displays to take those very weak sound waves deep inside the temporal bone. With a special sense of vision, we have to exert a similar kind of effort, this time to allow light to penetrate deep inside the body. Light is absorbed by all body parts. It is really rare to see transparent tissues. We'll see two examples of that and the examples of the eyeball. So special sense of vision creates its own effort to bring to the receptors those light rays that represent changes in the environment. In addition to what's happening inside the eyeball, we will spend a little time by describing a few structures outside the eyeball. So let me switch to screen sharing here. And what you are seeing is the first slide in these series of PowerPoint slides. Right, this is the first one. So while you see the eyeball right here, and light detection photoreceptors are all in there, plus many other structures that helps our quality of vision to be better. But we also have to have the ability to move the eyeball. And so to do that, we have skeletal muscles on the outside of the eyeball. This is a magnified version model of the eye socket. A lot of structures are taken out to show a clear picture. We see four of these rectus muscles. Here's the superior rectus, here's the lateral rectus. So on the foreside of the eyeball, superior, lateral, inferior, and medial, we have these straight muscles, the rectus muscles. Let me go to the next slide, which will show you the inferior and the medial rectus muscles. I had to take out the superior and lateral one. It clears up the picture a lot. In addition to these rectus muscles, which is just straight muscles, we have two muscles that grab onto the sclera sideways. Here's the inferior oblique, and here's the superior oblique. The superior oblique muscle takes a big turn right here at this point called the trochlea. And this muscle, through its tendon, will grab onto the sclera very much at an angle, just like the inferior oblique. Now, the action of these muscles, of the rectus muscles, is described by their name. So the superior rectus, when this contracts, it moves the eyeball superiorly. When the medial rectus muscle contracts, it will pull the eyeball medially, and so on for the other rectus muscles. The oblique muscles move the eyeball at an angle. The first point in the lab report says pair all skeletal muscles of the eyeball with the appropriate cranial nerve. So here's this little table. As you can see, two cranial nerves, Two muscles have dedicated cranial nerves to it. Lateral rectus has cranial nerve 6, and superior oblique has cranial nerve 4. It's okay to use the regular English numbers for 4 and 6. The same is true for 3. The rest of the muscles are all innervated by cranial nerve 3 or oculomotor nerve. So this completes this first segment of the lab report. Pair all skeletal muscles of the eyeball with the appropriate cranial nerve. In the lab report, there's a question of function or defect of skeletal muscles would affect the vision by what? So let me go back to this image. So the skeletal muscles allow our eyeball to track a moving object without the moving of our head. So that would not happen in this person where the skeletal muscles would be defective. Another effect that we get from the movement of the skeletal muscles is the angling of the eyeballs towards the middle. This is a natural position of the eyeballs. Through this positioning of the two eyeballs, we get the 3D vision and depth perception. So if these skeletal muscles would stop working, we would not have that 3D vision and depth perception either. The next part, and before I move on, we mentioned 3 cranial nerves. None of them are visible. They all bring nerve impulse to these muscles. But here's this large rope-like structure. It will be labeled later on, and it is also in the lab shell. This is cranial nerve 2. This cranial nerve collects nerve impulses from the retina and takes it towards the brain. So very different function for this highly visible cranial nerve. When it comes to the eyeball itself, we're going to see that the eyeball is made up of 3 thin layers of tissues, and these are called tunics. We can always come back to the slide. I will be describing these tunics by pointing them out on models, but this is always here. You're welcome to take notes on your lab report, of course, as we're going through. You can always check them against this slide. So on the outside of the eyeball, we have fibrous tunic. We have 2 components here. One is the sclera, whose function is protection of the eyeball, but to the anterior of the eyeball, this fibrous tunic becomes transparent. And so you can see this plastic shield-like material. This is the cornea, totally transparent. The function of the cornea is to focus light. Another word for that is refraction. If we take away the sclera and the cornea, we will be seeing components of the vascular tunic. The largest component of the vascular tunic is choroid. Choroid has 2 functions. It absorbs stray light from the middle of the eyeball, and it also brings nutrients to the retina. At the anterior part of the vascular tunic, we see this colored ribbon of the iris. The iris has smooth muscles, and the iris can regulate the amount of light that enters the eyeball. The iris does that by either constricting or dilating the pupil. The pupil is a weird structure in anatomy. It doesn't have any tissues associated with it here. It's just an opening, but it's an important aspect of the iris. Light rays are free to penetrate deep into the eyeball through this hole inside the iris. When you look at this model, you can see the iris here, you can see the pupil, you can see the choroid. It's usually described as a, or pictured as a purple material. A part of the, let me see if it's on the next slide a little better. Maybe I'm going to go do this first here. While it's not labeled, this flash colored part is also part of the vascular tunic. This is the ciliary body. The ciliary body has two parts to it. And when we look at the posterior aspect of the eye, here we see the ciliary body. It's a plastic ridge. The ciliary, on this model, the ciliary body has a ciliary process part to it. The process makes aqueous humor. And there is a smooth muscle, ciliary muscle, which changes the shape of the lens. So the lens, by the way, is surrounded by the ciliary body. Here's the lens in this model. And when we take the lens out, we see the backside of the iris, the posterior part. We see the ciliary body. We see the pupil. And this is probably best view to see the ridge the ciliary body forms around the lens. Two structures inside the vascular tunic had smooth muscles. One was the iris, and one was the ciliary muscle inside the ciliary body. Number 4 in your lab report is asking this: a functional defect of smooth muscles would affect the vision by what? Now, the smooth muscles of the iris change the diameter of the pupil. So whether we step into a bright light or a dim lit area, the iris would not change the diameter of the pupil. So we may be disturbed by high intensity light, or we may not see very well in a dim lit area if the iris smooth muscles were to be disabled. The smooth muscles in the ciliary body, the ciliary muscles, have the role of changing the shape of the lens. So this is necessary to see objects closer than 20 feet in a sharp manner. So if this smooth muscles of the ciliary muscles would be disabled, we would have no problem seeing any object further than 20 feet. But if you look at your cell phone, you would not be able to see the screen. It would be all blurry. We need to change the shape of the lens for looking at our phones. So with a disabled ciliary muscle, we would not be able to do that. We also have to say a few words about the lens itself. This is situated right behind the iris. The lens is very close to the parts of the vascular tunic, but it is not vascular, so it is not part of the vascular tunic, just located close to it. Again, the function of the lens is focusing light, refraction. The lens is the second transparent tissue in the eyeball. When we look at the third tunic, maybe I'm going to go back just once. Immediately underneath the choroid, going deeper, we see this very thin layer retina. This is also called the nervous tunic. And this is probably on a different model, is just painted on the inside of the model. You can see the purple color of the choroid out here. And right inside of that is the retina or nervous tunic. So we'll come back to these structures on the retina. claim to fame of the retina nervous tunic is that this is the site of the photoreceptors. In point number 5 on our lab report, let's name the structures of the eye in the order the light passes through them. And you can name a tunic in your lab report for each of them. So the first structure is the cornea. The second structure is the pupil. Light passes through here. The third structure is the lens. And the fourth structure that the light is passing through is the retina. And once light passed through the retina, that's when it's absorbed by the choroid. So retina is the last structure where light actually passes through. I'm going to move forward to talk about point number 6 in our lab report, dissection of the cow eye. So here is the dissection result of a cow eye. So the eye is cut into two cup-like portions. Here's the anterior portion, and here's the posterior one. Structures that can be recognized and might come your way on the practical is the cornea. Now, in this preservative fluid, the otherwise transparent cornea goes totally opaque. This is why it looks like this. You can see the sclera right here. So it's another structure you can observe. The ciliary body forms this ridge, just like on the model. The lens was taken out. The lens would be sitting in this depression, but the lens is also opaque because of the preservation fluid or preserving fluid. So here's the lens. You can see the retina in the posterior segment of the eye, the cow eye. It's hard to see, actually, let me just put it this way. This cup posterior cavity of the eyeball was filled with a gelatinous liquid called vitreous humor. This is crystal clear, and that pressed the retina against the choroid. And you can see here the choroid, pitch dark layer, the posterior aspect of the eye. Now we see this blue layer here. This is not present in human eyes. This blue layer is called tapetum lucidum. Tapetum lucidum reflects the light inside the eyeball. This is very useful for nocturnal animals like deer, cow, other ruminants. Human eyes don't have this blue layer, so you don't see this labeled here. So these are the structures that you're expected to recognize and name and associate with a function coming from the cow eye. The next point in our lab report is asking you to take notes on the monkey eye cross-section. So let's see if we can go there. I'm sorry, I need to stop sharing here and switch to the lab shell. Let me see if right here. So under the microscope at a fairly moderate magnification, this is what we see if we cut the eyeball along a sagittal plane. So here is the anterior portion of this monkey eyeball. Here's the cornea. Here's this space that we will be naming very soon. You can see the iris. Immediately behind the iris is the lens. And you see a big space here posterior to the lens. The ciliary body is illustrated here separately, pointing at the ciliary process and the ciliary muscle. So in addition to recognizing these structures, it's important to name these spaces. These spaces are all filled with a liquid. So posterior to the lens, The space all the way to retina is called the posterior cavity. Posterior cavity is filled by vitreous humor, this jelly-like, very clear substance. Anterior to the lens is the anterior cavity. This has two chambers. Between the cornea and the iris is the anterior chamber. And between the iris and the lens, this little sliver of space is called the posterior chamber. Both chambers, and therefore the entire anterior cavity, is filled with aqueous humor. As you can see, aqueous humor, as we mentioned, produced by the ciliary process. This liquid produced here can move between the lens and the iris, and then through the pupil come out into the anterior chamber. So this is the summary of parts that you can see on this monkey eye cross-section, and we added to that the name of the spaces and the liquid types that fill out those spaces inside our eyeball. So for the retina, before we go into details of photoreceptors and their properties, let's take a look at another microscope slide. This is, again, I'm projecting from a lab shot. This is the cross-section of the retina. Actually, there's a lot more here besides retina, but that's probably the most important aspect. As the eyeball situated on this microscope slide, the light would be coming from a top, and the light would be penetrating through different layers of the retina. So let's go through this one by one. Light hits the retina at the ganglion layer. This is a whisper-thin layer of cells. These are neurons. Light just passes through them. The light then hits the bipolar layer. This is also a layer of neurons, much thicker, as you can see, thicker purple band here. Light passes right through and hits the photoreceptor layer. We'll come back to this layer. This is where we find the rods and cones. But lo and behold, light passes through this layer as well. And so now you see the strategically positioned choroid. Light does not pass through the choroid layer. Choroid is so pitch dark, it absorbs the scattered light from inside the eyeball. If the choroid wasn't at this location, sclera would reflect the light back through the retina again, and light would be bouncing around inside the eyeball. So this is where really important property of the choroid absorbs stray light. Anything that passed through the photoreceptor layer is being absorbed right here. So the photoreceptor layer has these receptors, rods and cones. And when you go to the third page of the lab report, here's number 9, photoreceptors of the retina. So let me go back to these PowerPoint slides that will be attached to your recording. And let me see if I can switch screen sharing back to this. Right. So the photoreceptors in the nervous tunic or in retina, we have the rods and cones. Now we cannot distinguish these on our microscope slides, so this is why we just type this out. Rods detect shades of gray, cones detect color. Not only that, but the cones are highly enriched in their density in this area of the retina called macula lutea. On the flip side, there are no receptors in another part of the retina called optic disc blind spot. So when we look inside the retina or the model eyeball. We see the retina, and here is the macula lutea. This is a small area. Again, cones reach highest density. A consequence of the high density of cones is that the best visual acuity or sharpest vision, and the macula lutea is also responsible for the best color vision. No surprise that the image that we are focusing on anytime always projected onto the macula lutea to take advantage of the high density of the cones. The ganglion cell layer bundles axons of those neurons into this large area. This is the optic disc or blind spot, and the photoreceptors are shoved aside by those axons, so there are no photoreceptors, neither rods nor cones, in the optic disc or blind spot. So this is how these areas show up on the model in the lab. So when we named these two different areas, macula lutea and optic disc, we were talking about points number 10 and 11 in the lab report. Here we are beginning to talk about point number 12. Now, for this, you need to go to your lab manual. And on page 378 and 379, at least in my manual, these are the pages, you see a number of visual tests or vision tests illustrated. On page 379, you see a chart with large numbers decreasing in size. This is the Snellen eye chart. This is also used in a much smaller version as Snellen acuity card. So in our lab report, number 12, perform the vision test to detect nearsightedness. This is the distant visual acuity test. This is described in page 378. You're looking at this Snellen eye chart from a 20-foot distance, and this can detect nearsightedness, meaning issues with visions further than 20 feet away. Number 13 is using a different tool to detect farsightedness, and this is the tool that you see here. This is the near visual acuity test. This is where you see the Snellen card. And this tool allows you to put one end of this tool just below your eye. For vision tests, you always have to close the untested eye. So leave this eye open and slide this card closer and closer to your eyeball. When you see the Snellen eye card being blurred, that's the distance of this card will tell you the near visual acuity distance. In order to perform this test, your lab report is asking which part of the eye changes shape during this test. Well, our lens needs to become more and more curved as the Snellen eye card is approaching the eye. So it's the lens that changes shape during this near visual acuity test. We also have a test of color blindness. This is point number 14 in our lab report. Because only cones detect color, color blindness test is designed to detect malfunctioning of cones, the one type of photoreceptor. What you see here illustrated is a set of Ishihara tables This is a classic tool to determine color blindness. And again, this set of panel and many more in this booklet is testing functioning of the cones. Actually, before we move into the other special senses, let's talk about point 15 here in the lab report, astigmatism. If you look in your lab manual on page 379, you see an illustration that looks like a circle in the middle and spokes of wheel attached to it radially. So this is the astigmatism chart. And looking at this chart from more than 20 feet away, the astigmatism test detects imperfect curvature of either the cornea or the lens. So anybody with an imperfect curvature of the cornea or lens would not see a symmetrical image. These spokes of wheels, these lines may be squiggly. The central structure might not appear to be a circle, but an oval, or have distortions to it. So this astigmatism chart on page 379 detects the uneven curvature of the cornea or the lens, and that disorder is called astigmatism. For the last part of this lab, we're going to spend a little bit of time on special sense of smell and taste. So we don't have any models or microscope slides at this time. Let's talk about special sense of smell. This is also called olfaction. Olfactory receptors detect chemical change in the environment, whatever is floating around in our nasal cavity, chemicals in that air are detected by olfactory receptors. These receptors, olfactory receptors, are located in a saran wrap thin tissue called olfactory epithelium. This is located in the top region of our nasal cavity, and this thin tissue is supported by the ethmoid bone. Once olfactory epithelium and receptors detect changes in the chemical environment in the nasal cavity, let's say you're smelling a freshly opened banana, or you are detecting a new car smell, or if you are detecting the smell of a gas leak, nerve impulses will be created, and it's the olfactory nerve or cranial nerve 1 that carries those nerve impulses all the way to the brain. So this is all we have about olfaction, special sense of smell. We have a little more about special sense of gustation. Sorry about that. So the taste receptors are located in taste buds. The taste buds, in turn, are located inside these fairly large structures on the surface of the tongue called papillae. And taste buds are in the valate and fungiform type of papillae. We have no taste buds in the filiform papillae. The filiform papillae give us tactile sensations from our tongue. We can detect the texture of a food. If you have a cherry pit in your mouth, you can find that very easily just by fishing around in your oral cavity with the tongue. The filiform papillae allow you to detect that cherry pit and anything solid in the oral cavity. Number 18 is about in the lab report, naming the 5 taste receptor types and the chemicals they detect. So here they are listed. So please take notes in your lab report. Sweet receptors detect simple sugars. Salty receptors detect sodium ions. Sour receptors detect hydrogen ions. And bitter receptors detect small organic molecules. Think about small organic molecules like the molecules in roasted coffee, the hops in beer, just two examples of bitter tasting materials. Umami receptors detect amino acids. So those are the taste receptor types with the chemicals they detect. And the last item here in the special sense of taste or gustation, that taste-related nerve impulses carried to the brain by cranial nerves 7 and 9. And these are facial or and glossopharyngeal nerves. So this is the recorded version of this second lab. I hope you'll find this useful for your preparation of practical one. Again, you need to have a lab report, a lab manual, and of course, I was projecting the microscope slides from the lab shell. Please look that up and other illustration there as well. I'm going to stop screen sharing here. So this is the end for this lab. If you have any questions, please send me a message through the class shell. And I'm going to stop recording here and send this on your way.